We're back. Plenary session, unedited monologues. This is what we're going to do in season 4. It's hashtag zero COVID. We're not talking about COVID. And if you go outside, no one's talking about it. It's back. It's back. People are back and we're back. We're back on oncology. So far this season, we've had some great interviews. George Sledge, Nina Shaw. We're going to have more interviews coming up. I don't want to spoil it for you. But today I have a few things to talk about. I want to talk about a little thing I saw on Twitter. And let me share my screen if that's doable here. I saw this. This was by Graham Collins. This is a slide that he saw from, I guess, a talk he went to. And this was, I thought, a little interesting management. An approach to high tumor burden follicular lymphoma 2021. BR frontline, sure. We've got some randomized data there. Followed by Revlimid rituximab. Oh, that's a great study because you got Revlimid rituximab randomized against rituximab. Yeah, not exactly what I like to see, because obviously if somebody can tolerate Revlimid rituximab, I wouldn't be giving them rituximab. I'd be giving them something stronger. Of course, if you're fit, then you get CAR T. But if you're not fit, then you can think about tazemetostat. Sure, if they happen to have that one mutation. And then, of course, everyone trickles down to the PI3 kinase inhibitor. Okay. I saw this. I don't know what the purpose of this slide is, but I will say a few things about this topic. This is not good. With follicular lymphoma, you don't need to be giving all of these drugs. They don't have credible evidence. We have some PI3 kinase inhibitor studies in combination with rituximab versus rituximab placebo. Is that acceptable in follicular lymphoma? I think not. I think we want to see that against an actual control arm of our chemotherapy. Revlimid rituximab R squared, does it have activity? Sure. Activity means it can shrink tumors in some fraction of people. Does it have efficacy? Is it better than alternatives? And the answer is I have no clue at all, because the only data I have are the flawed randomized trial against placebo rituximab. What about that tazemetostat? I have uncontrolled data. What am I to do with uncontrolled data from a novel, costly drug? And what about CAR T in the fit? Again, Zuma 5, uncontrolled data. I look forward to randomized data. I got a lot of problems with this space. We have a standard of care for a reason in lymphoma. That reason was not always randomized trials that showed every step of the way was beneficial, because this was developed in the 1960s, 70s, and 80s. They naturally used the standards of evidence of that time, which often was uncontrolled studies. But now we're in 2020. We're in 2021. We're in a time where the standard of care should only change based on randomized controlled trials showing superiority. The thing about the old studies was, back in those days, the cost of a cancer drug wasn't 4 times the average household income, okay? These were cheap drugs. So you could get, and also the knowledge of science also changes over time. You know, they didn't do randomized controlled trials in the Stone Age. Does that mean we should never do them today? The standard of evidence gets better over time. So for those reasons, I think you ought to test these drugs against the best available standard of care, which is our chemotherapy in follicular lymphoma. If it's been long enough, you can give them BR again. You can give them RCVP. You can give them R-CHOP. These are all regimens with activity. If they're frail, you can give them R-cyclophosphamide. You can give them R-chlorambucil. You can give them all these drugs. You don't need to get into R squared, tazemetostat, PI3 kinase inhibitor. You can save those for lines beyond these lines. Of course, we know follicular lymphoma, sometimes when it progresses, it's transformed, in which case we're on a different pathway entirely. I don't know what to say. I think it's a shame that we permit these drugs very broad marketing authorizations based on evidence that is quite flimsy. Somebody was arguing with me online and they said, well, you know, the thing about Revlimid rituximab versus rituximab is the control arm is fair because a lot of people can only tolerate monotherapy with rituximab. And I wanted to say, sure, that's plausible in some people's minds, not in my mind, but I'm sure you could argue that. But in this algorithm, you do note that after they have exhausted R squared, they get CAR T therapy. So you mean to tell me that you got somebody so fit they can get CAR T therapy, but they couldn't tolerate anything more than rituximab? Get out of here. Get out of here. You're not fooling me. We all know why the companies want these lackluster, subpar, unethical control arm studies. And that's because they get the market share. It's up to the doctors, the trialists, the sensible people to say we need control arms that actually reflect what's going on in the world, which is our chemotherapy. To be perfectly honest with you, if I had none of these options, I'm not sure patients would be any worse off. I have no credible evidence that any of those options is superior to just giving different types of chemotherapy. We don't have any good studies in this space. Isn't that a shame? Isn't that an embarrassment for follicular lymphoma? So when I saw this, I chuckled and I said something like, this is like how you'd make an algorithm if it was designed by the pharmaceutical industry. And ironically, it's probably going to be that way very shortly in the NCCN because that's almost an extension of that. Um, One, because as Aaron Mitchell and colleagues pointed out, that many NCCN authors have financial conflict of interest with the industry. I would think it was 85% with a mean payment of $16,000, which is more than your average oncology oncology physician. The second piece of evidence is that the NCCN often extrapolates beyond available data to make their drugs more, to make novel, costly drugs be given to more people, often based on low levels of evidence or no evidence at all. And the citation for that is the paper I did with Jeff Wagner in the BMJ, Frequency and Level of Evidence of the NCCN Recommendations. I think we're in a bad place. We're in a bad place in lymphoma where you're starting to approve many, many drugs based on either uncontrolled studies or studies with bad control arms. And then you create algorithms where you don't give any of the other regimens that we are used to giving, and instead give these more costly regimens without ever having proven superiority to what we're actually doing. Standard of care is a slippery beast in biomedicine. It may come into being based on all sorts of reasons, including historical accident, uncontrolled studies. But once standard of care is in place and you do a randomized trial, you have to randomize control arm patients to the standard of care. You can't just give them placebo rituximab saying, well, the evidence wasn't that great when you developed the standard of care. Imagine if they did that for EVDs or VP shunts or appendix removal. Those are all standard of cares developed for in a time where we didn't routinely do randomized controlled trials. So my position is the following. Randomized controlled trials are the best way to assess if novel therapies are superior to current standard of care. But the second part of my principle is that when you do those studies, the control arm has to receive the standard of care. They can't receive subpar care, even if that standard of care was not itself supported by randomized controlled trials, because it was developed in an era where we didn't require that routinely. So medical evidence can get better. The standards of evidence should get better. And just because they didn't do it in 19 diggity doesn't mean we can't do it today. So those are my thoughts on this topic. Next topic, ivermectin. This is zero COVID, so I'm not going to talk about ivermectin. I'm just going to say a couple things because it has relevance to oncology. I got sucked into this reading this meta-analysis on ivermectin, and I have nothing but regrets, as one would when one gets sucked into reading a meta-analysis of bad randomized controlled trials and beyond observational studies. A meta-analysis of observational studies is like a septic tank. It's nothing good that comes from putting all that together. It's no good. I don't know what to say. We're going to talk about this in the last part when I'm going to take you through a new paper of mine. But what do I want to say about this? When I looked at this, I noticed a few flaws with the data set. One, they were extolling underpowered randomized controlled trials that were not designed or suited to find survival differences that happened to find very large survival differences. That's bad. If your primary endpoint is not survival and you do a 50-person randomized controlled trial and find a 20-month survival benefit, that's very likely to be spurious. We all know underpowered studies mean you're prone to false negative results, i.e. that you failed to find a difference when in fact a difference existed. But what is underappreciated is that severely underpowered studies are also prone to, when they do find positive results, that those results are more likely to be exaggerated and or spurious. And if you want to see the mathematics behind that, there's a paper, I think, in one of the Nature journals called Power Failure, and it was about neuroscience. It'll run you through the equations that prove that to be the case. So when I looked at this meta-analysis and I saw that people were praising underpowered randomized trials showing ivermectin works, I thought to myself, shoot, what about POLA-BR? People say Pola BR has a survival benefit over BR. What they don't realize is that that's an underpowered phase 2 study, and that's an accelerated approval. And we don't know if that finding is true. Let's replicate that study with some real sample size that can actually assess survival. In fact, I suspect it's almost surely exaggerated, and it may be spurious entirely. We will see. I also thought of olaratumab or Lartruvo, that soft tissue sarcoma drug. Well, that's a great example. Underpowered phase 2 study find a huge, almost incredulous OS benefit. And then the phase 3 randomized control trial found superimposable OS curves. So, we're critical of ivermectin for being, I think, borderline conspiracy theory, low-level science. But look, we got 2 examples in our own field. And what about Saber Comet or all these oligometastatic RCTs? Where's your power calc? Where's your phase 3 study? You have already implemented this based on phase 2 studies that are not designed or suited or powered for OS. Let's do a real study with adequate power. So that was one thing that jumped out at me when I looked at it. The next thing were anecdotes. Somebody said that, they had somebody on the vent and they gave him some ivermectin, and all of a sudden their lab started to correct and things started to go in the right direction. So, we don't need randomized trials. We have experience. What could be better than experience? Well, of course, experience is misleading, deeply misleading. And I also saw a parallel to oncology. I hate to say it, my friends. We give tocilizumab in CAR Ts. Where's your randomized trial of Tosi? Where's your randomized trial of anakinra? That's the new one. Anakinra. We'll give sub-Q anakinra. Why don't you do a little bit of a randomized trial and let's see what these actually do versus merely the natural course of some of these conditions. I think those are important studies to do, and the apprehension of doing them, I think, is not so good. What about the patient who we refer to molecular sequencing, and then we find out that they have a TSC1 mutation? We give them everolimus, and let's say their tumor doesn't shrink, but we think that it slows the growth. So-called precision oncology or molecular tumor board panels. Is that any better than ivermectin? Telling some story, some fable about what you think you saw, what you thought you experienced? I don't think so. And then finally, what about aducanumab, the Alzheimer's drug? Oh, this is a great drug, right? Obviously, I've never seen so many people universally crush a drug in the op-ed pages. But the bottom line is Biogen ran trials that either failed to accrue, failed to meet the primary endpoint, or otherwise negative. They change amyloid protein, which is a poor and unreliable surrogate. In fact, even calling it a surrogate seems an overstatement because we have little credible evidence that altering it actually changes the condition. Yet we get an approval for Alzheimer's, all comers, which is potentially 6 million people at $56,000 a pop, which if you assume 1 in 3 people actually get the product, you're talking about a budgetary impact of $112 billion, which will crush us, a crushing budgetary impact. What's my point here? My point here is that ivermectin, it turns out YouTube censored videos that extol ivermectin. Am I a fan of ivermectin? No. I think ivermectin needs a large randomized trial or STFU, or keep quiet. I don't want to waste my time with ivermectin. I think a large randomized trial, if I were to put money on it, it will be negative. And why will it be negative? Because most things in biomedicine don't actually work. We have low pretest probability, particularly of drugs. Ivermectin will have a low pretest probability in severe illness and prophylaxis and all these things. And if you do a really clean, elegant, large randomized control trial, it's going to go bust. People said, well, there's no money to do that. It's a conspiracy because nobody gets rich off ivermectin. Well, guess what? There's no money in the big dexamethasone business, but they actually did it there. So spare me the excuses. But my point is this. YouTube has now thrown their videos off the platform. I'm happy to let them keep their videos on the platform because, YouTube is full of garbage. Let's be honest. But what I think the crux of the issue is, what about aducanumab? The evidence for aducanumab and the evidence for ivermectin are both very low quality. In fact, probably ivermectin, maybe even when you get that low, let's not actually make comparisons. They're both in the garbage section of the evidence base. But yet aducanumab very likely will be advertised on YouTube as advertisements. So YouTube is a very interesting, I think, technology company that actively bans you talking about one unproven drug while probably running advertisements for many other unproven drugs. One happens to have the FDA stamp of approval, but if anything, doesn't that lend itself to the credence of these conspiracy theorists who think that if there was a way to make money off ivermectin, we would have gotten an accelerated approval? Maybe. I think there's a bigger failure here, which is that we do a very lousy job of communicating medical evidence. That lousy job translates into some things in the Western accepted medical canon, like your molecular tumor board. I urge people who run these molecular tumor boards, randomize 500 people to your molecular tumor board or send them to a clinic where a good doctor like myself thinks about what to give him. And maybe we give him some etoposide, some Cytoxan, some platinum, and measure overall survival. And in fact, I did the power calculation for such a study. It's in the Annals of Oncology. It's called Why F1CDX Should Get a Randomized Trial Before Medicare Pays for It. I did that power calculation, so I can tell you exactly how to do that study. I suspect your molecular tumor board may fall short because I think even with old cytotoxic drugs in salvage settings in solid tumors, I can easily match you or beat you in OS. You can do whatever sequencing you want and give them whatever pazopanibs and imatinibs and whatever the hell you want to give them. But I bet I'll win because I don't have to waste my time waiting for tumor board results that are inconclusive. I don't have to bet the farm on some targeted TKI that's actually quite intolerable when you dose it daily. And I can deliver my drugs very quickly. But you've never done that study. You've never done that study. And yet you continue to have your molecular tumor boards. The ivermectin people are not doing a proper randomized trial there, but aducanumab didn't. So I think we have a real crisis when the bar between FDA approved and the topic of a conspiracy theory is a very narrow distinction between those two. The answer, of course, is clear. Everybody needs to produce evidence or they need to stop making claims. And so claiming that R squared is better than R chemotherapy is an unfounded claim. Claiming that we can't randomize people to anything other than R versus R squared is unfounded if your pathway shuttles them to CAR T thereafter. If they can tolerate a CAR T after they've progressed, surely they could have tolerated something more than rituximab. So evidence is this very interesting thing. It's a tool. It applies to both canonical medical practices as well as alternative, complementary, fringe medical views. And you need a framework of evidence that has some consistency. And when I look across these spaces, I see very little consistency. Pola-BR, that has FDA approval. Well, by that logic, then some of these garbage small randomized trials of ivermectin, maybe they should get FDA approval too. I disagree. They shouldn't, because that's an underpowered study. But then why the hell did Pola-BR get it? Why did olaratumumab get it? They shouldn't get it either. Okay? So we need to do some soul searching. We need to appreciate medical evidence. We need to understand medical evidence. We need to understand why large randomized trials are the best, which I'm going to show you in the third part of this extemporaneous monologue. And we need to have clear standards. I'm happy to tar and feather the proponents of ivermectin, but I also want to tar and feather the proponents of aducanumab too. They're all the same in my view. They're people who try to foist upon society useless things that don't work. The ivermectin people, at least they're cheap. You people are going to bankrupt us. So actually, I don't know what's better or worse. I don't like to split hairs when we're in the garbage can of medical evidence. Okay, the last part, the new paper I have. Where is this paper? Oh my gosh. Okay, now we're cooking with gas. Okay. I hope you can see my screen because now there's literally nobody out there to tell me. And that's also a stupid cliche thing that I've heard too much this year. Okay. Go to my website, vinayprasad.com papers. When I talk about a paper, this is the place you want to go look for it. And here's what you'll find. Reliable, cheap, fast, and few. It has a link. This is the new paper out. It's in the European Journal of Clinical Investigation. Okay. I want to talk about this little paper because it piqued my interest. And I want to show you a couple things about it. I got this idea last summer, and I thought to myself, there's really three major types of study design. There are randomized control trials, at least when we talk about the efficacy of therapeutics. There are retrospective observational studies, and there are synthetic or historical control arm studies. This is the new frontier. Everyone's getting all excited about synthetic control arms. What are these studies? What are their advantages, disadvantages? It's all in this paper, but let me walk you through it. One, reliability. What you want more than anything is you want reliable causal inference. You want to know for sure this therapy does what you think it does. That actually comes best from large pragmatic randomized control trials like Recovery, like TASTE, like Stampede, very large studies with very low inclusion criteria. You include everybody, average people in your country, and then you sort out whether or not randomized to this intervention or not this intervention, you have a benefit. Okay, that's reliable. Randomized controlled trials are hitting the ball out of the park when it comes to that type of internal validity. When it comes to reliability, when they make them pragmatic, I think it's the strongest truth claim in all of biomedicine that something actually works. What about observational propensity, score, matched, weighted, inverse probability weighted kind of studies? The answer is they have some concordance with those large, well done randomized controlled trials, but the concordance is not infallible. There is a failure rate. And in the paper, I describe that failure rate is asymmetric, that these observational studies are much more likely to conclude that an unproven therapy that's toxic and invasive works rather than an unproven therapy that's toxic and invasive doesn't work. Let me put that another way. When they find these observational studies examine practices, they find when these observational studies claim something works, it mostly doesn't work. When they claim something doesn't work, it mostly doesn't work. Doesn't work is the key right answer most of the time, but the observational studies insert the illusion that tends to say that more toxic, more invasive things work. And that's probably confounding by indication, i.e., these things are being deployed in healthier people, which is hard to separate from the effect of the therapy. And thus it gives the illusion that the therapy is actually doing something when it is, in fact, a selection bias for that therapy. I think that's the bulk of it. So that's the problem with these retrospective observational studies, these NCDB studies. You can read the paper for some examples. I've just given you a few. But what about the other parts of the argument? That's reliable, cheap, fast, and few. Cheap and fast. This is interesting to me. We want this to be cheap. We want to find these answers without spending as much money as possible. And randomized trials, they're damned expensive, I keep hearing. They're $50,000 per person. Who the hell can afford these randomized studies? Well, it turns out if you don't do the randomized studies and you just deploy interventions, you're going to have to do it to a bunch of people before you have enough people to analyze for your observational study. And how many people is that? Well, the answer is in a pooled analysis of 2 studies I look at, it's anything from 1 to 6 to like 1 to 8. In other words, 8 times or 6 times as many people have to be subjected to an intervention if you just deploy it before you run your observational study than if you had randomized. And that should be natural, right? If you deploy a treatment, a lot of people are going to gain access before you have time to assess whether or not it works. And that lot of people is probably far more than the power calculation of a randomized control trial. So in this paper, I did something, well, that I thought was interesting. I don't know if you think it's interesting, but I think it's quite interesting, which is I did some math, which apparently I still remembered how to do, perhaps not so well. But basically, it says if you assume this cost per participant in an RCT, $50 per person, which is what they achieved in TASTE, 1,000, 20,000, 50,000, which is what I've highlighted in bold. This is the price per therapy where it's actually cheaper to do the randomized study if the therapy costs more than X. In other words, if it's $8,000 or more, it's cheaper to do the RCT. Now, why is that the case? It's the case because even if you account for a $50,000 premium per person in an RCT, you would have had to treat so many more people in the community that $9,000 medicine would actually cost more if you just deployed it in the community than if you actually did the RCT, giving it to fewer people, but paying $50K per person to be in your study. And that's, I think, a sort of on the high end figure. But it does remind us that most things in oncology, we're not talking about $8,000 medicines, and that's for the total cost of the thing. We're talking about $400,000 a pop or $200,000 a year. So most things in oncology are actually cheaper to assess in a randomized fashion than a target trial or an observational study because you have to give it to far fewer people before you sort it out. Fast and few, randomized trials answer questions rather quickly. And I give some examples in the paper of when we didn't do them, how long it took to get the answer. And few randomized trials only are they limit to some degree the number of people who are provided an inferior therapy. That is either the losing arm of a randomized control trial, but that number of people in the community, if you just deploy things, might be far, far bigger. And you'll have to read the paper to see how I think about that. But what's my take home here? My take home here is that there are many people who want to talk about the limitations of randomized trials. Sure, I'm one of those people. In fact, the book Malignant gets into a lot of the limitations of randomized controlled trials. Just because you're a randomized study doesn't mean you're running a good randomized study. But in contrast, if you are making a therapeutic claim of efficacy in the absence of a large, adequately powered randomized trial, you are making a somewhat unreliable claim. The degree of unreliability is linked to the degree to which your claim falls short of a well done, adequately powered randomized controlled trial. Aducanumab, sort of a tenuous claim. Ivermectin, tenuous claim. Olaratumumab, tenuous claim. Pola-BR, tenuous claim. Oligometastatic radiation, tenuous claim. You don't have adequately powered randomized studies. That entire algorithm for follicular lymphoma, tenuous claims. You have bad control arms or no control arms at all. So you suffer from reliability. That's what randomization gets around. You actually, ironically, might even increase cost. Low regulatory hurdles will just flood the market with very costly drugs. And if they cost more than $8,000 per pop, it probably would have been cheaper to do a randomized study. $8,000 per pop is actually far lower than the majority of things we do in oncology. There are not too many things we do cheaper than that. So most things in our line of work, not only is it more reliable to do the randomized study, it's actually cheaper too. And that's the key point of the paper. I think that's the key insight of the paper. So we've been talking a while. This is the new plenary session, #0COVID, which means I have no continuing interest in something that is dwindling. And we're going to talk about oncology. And I hope we did that a little bit here. I'll be back in future episodes. I'm going to take you through this immunotherapy paper about fecal transplant immunotherapy. It'll be a good one. And I'm looking for pivotal randomized controlled trials to talk about. I think I talked about a couple in our ASCO roundup, and we will talk about more in the weeks to come. And I'm going to try to book some of the luminaries in oncology to get you their perspective. The last thing I would say. Some people ask me why you don't argue more with the people on your show. And I guess I would say for a couple of reasons. But the biggest reason is that I would just feel bad about it. I mean, I don't know. I invite someone to come on this show. This is a very modest enterprise, let's be honest. If I invite them just to argue with them, perhaps in a strong way, and if I make them look poor, I think that's a poor reflection of the host. And so it's just merely a sense of etiquette that keeps me from it. But my views are very clear. The goal of cancer medicine is to improve survival and quality of life. The goal of very costly drugs is to know with certainty or some confidence that you do that. And there are ways to do that that require better appraisals of evidence. You need to do randomized studies. When you do randomized studies, the control arm needs to be what you'd otherwise do. The post-protocol therapy needs to be what you would otherwise do in both arms. Sometimes if you're moving a therapy from last line to front line, you need crossover. And if you have a therapy that's never proven benefit, you don't need it. And you can check out the YouTube videos on clinical trial appraisal to learn about this crossover issue that Allison Haslam and I first defined in the Annals of Oncology a few years ago. When you are doing this N of 1 personalized oncology, it is infeasible to do a randomized controlled trial for every mutation you find. But you can do a randomized controlled trial of the strategy. And the most important point here is that whether something is Western, canonical medicine, or fringe alternative medicine, evidence is the same. And if we are to be critical of ivermectin, even lampoon the people who promote it, we should also be critical of some of the things that we have embraced. Pola-BR. Pola-BR, that study is just too small to have any strong OS benefit. If you doubt that, look at Lartruvo. It was the exact same thing, but they did a follow-up study. Oligometastatic radiation, it's nothing but small phase 2 trials. Do the large phase 3 trial. We need some consistent framework, and I will hope to define that in the weeks to come. So on that positive note, we're done. Short episode, plenary session. We'll be back with more interviews. Good ones coming. Stay tuned.